Hello, my name is Marney Pratt. I'm a senior laboratory instructor in the Biological Sciences Department at Smith College. Today, we'll be talking about my experiences over the last year doing authentic research in the undergraduate classroom, comparing the impact of land use on macroinvertebrates and water quality. There are a lot of benefits to course-based undergraduate research experiences, or CURES, such as improvements in science practices and lab skills, content knowledge, confidence, and attitudes and interest in STEM careers. There are many ways to include research in the education of undergraduate science students, from verification labs following a specific protocol to completely independent research. My course falls more towards the open ended research, within parameters set by the instructor. In this case that I'm talking about today, students did not design the study or collect the data themselves, but they did get to choose their own question of interest to answer with a huge amount of data available to them. The course I'm talking about today is Research in Biodiversity, Ecology, and Conservation. This is an introductory level course with no prerequisites. I usually teach 2 to 4 sections each semester with a maximum limit of 16 students per section. The course has a wide variety of students, including biology majors, environmental science majors, lots of other sciences, students majoring in other sciences, as well as non-science majors, really a wide variety of kinds of students. During remote teaching this year, we used data collected by me and various Smith College students and colleagues from the Mill River, as well as data collected by the National Ecological Observatory Network, or NEON, from the Lower Hob Brook. Both rivers are wadable streams in western Massachusetts, only about 40 kilometers apart, and experience a very similar climate. The sampling was not done exactly the same way, but I think it was similar enough to give an interesting comparison. I will present data today on the macroinvertebrates and several water quality measurements. pH and dissolved oxygen were measured in the Mill River during macroinvertebrate sampling in the same locations that the macroinvertebrates were sampled. However, all the Lower Hop Brook water quality measurements were made from stationary probes that were measuring water quality continuously in one location. In the Mill River, we also had a turbidity and water temperature probes that were in stationary spots that measured continuously in that same location. In the case of the pH and the dissolved oxygen, I just matched the precise time and day that I measured things in the Mill River to those same days and times and measured in the Lower Hop Brook. These maps were made using modelmywatershed.org. They show the watershed for each river outlined in white. The rivers are blue, so here's the Mill River, here's the Lower Hop Brook, and the point at the lower right is the point where sampling was done. The different colors represent different land use. Note that the Mill River is a much larger watershed at around 140 square kilometers, whereas the Lower Hop Brook was just over 12 square kilometers. The Mill River has more developed areas, shown in red, whereas the Lower Hop Brook tends to have a higher proportion of forested areas. The students hypothesized that the Lower Hop Brook would have greater macroinvertebrate density and greater water quality than the Mill River because of the differences in land use, the Lower Hop Brook having more forests and less development. Now I'll show you a bunch of results that we found from doing this comparison between these two rivers, and they'll all have a similar general format where we show each point represents a particular sample in each river. The different colors are from the different rivers, and the shape of is showing the distribution. So further wider out of points means there were more points at that value. So in this case, like around 1. There was a lot of values of turbidity around 1 in the Lower Hop Brook. I also show the mean is an X and then a box plot, which shows the median as the middle thicker horizontal bar. The box itself is the middle 50% of data, or the interquartile range. And the whiskers represent, in this case, the lower whiskers reach to the minimum point, and the upper whisker reaches 1.5 times the interquartile range from the upper hinge of the box. Anything outside of the whiskers are considered outliers. Because the turbidity data really was, there's a lot of low values and then a couple high values, I have graphed it on a log scale in this case. And each value in this particular figure shows the mean daily turbidity, so the average turbidity on a particular day. And what we can see here is that the variance, the mean, and the median were significantly greater for turbidity for the Mill River. So generally, while there's a lot of overlapping points, in general, there's higher values of turbidity in the Mill River than in the Lower Hop Brook. Water temperature, shows the mean daily water temperature on a linear scale in this case. Water temperature also showed greater variance, mean, and median for the Mill River. So in general, the temperature of the water is warmer in the Mill River. We also looked at this on, over time. So these points, again, are daily values, the mean daily water temperature, but for each day from 2018 to 2021. And what I want you to notice is that during much of the year, there's a lot of overlapping points. They're not hugely different, but the biggest differences happen in the warmer months. So you see a bigger difference between the two rivers with warmer water in the Mill River in the warmer months. Dissolved oxygen showed greater dissolved oxygen in the Mill River than the Lower Hop Brook, the mean and the median being significantly greater, but the variance was greater for the Lower Hop Brook. And this is a linear scale, and this is a surprising result. We didn't expect to find higher dissolved oxygen in the Mill River. We just expected to find higher in the Lower Hop Brook, but we found opposite of what we expected. And I also don't know why there's this much lower clump of data in the Lower Hop Brook. Because temperature affects the dissolved oxygen, I also looked at dissolved oxygen as a function of mean daily water temperature. This was an analysis that I thought of, not the students. So this is just my own analysis. But you can see that at the same temperature, we still have greater dissolved oxygen in the Mill River than the Lower Hop Brook. We would expect for really nice, clean water and fresh water, the pH would probably be fairly close to 7, which would be perfectly neutral. And the Lower Hop Brook is pretty close to 7. It has a lower pH, but on average, the variance, the mean and the median, were significantly greater for pH for the Mill River. So the values in the Mill River vary more, and they are a higher pH. So that could be a potential stressor for certain organisms that prefer a pH closer to 7. Moving on to macroinvertebrate density, we see that the variance, the mean, and the median was significantly greater for the Lower Hop Brook. And do note that this is another scale that's on a log scale, because there was quite a bit of variation between the different samples on what the macroinvertebrate density was. But while, again, there are some overlapping points, generally speaking, there are more invertebrates per unit area in the Lower Hop Brook than the Mill River, which is what we predicted. But again, because they were not sampled exactly all on the same dates, I wanted to compare the macroinvertebrate density to date. And you can see even dates that are very similar or close, the Lower Hop Brook still has much greater density than the Mill River. So the difference between the two locations isn't just explained by the differences in sampling date. Oops. Another value that we compared is the tolerance to pollution. So each kind of organism was scored on whether it was really tolerant to pollution, closer to 10, or very sensitive and not tolerant to pollution, closer to 0. And then these values were put together basically in a weighted average to give the Hilsenhoff Biotic Index. The mean and the median of the Hilsenhoff Biotic Index was significantly greater for the Mill River. There are a lot of overlapping points because we do have a lot of similar kinds of organisms found in the two rivers, but we are seeing a little bit higher HBI or Hilsenhoff Biotic Indexes for the Mill River than the Lower Hop Brook. So our conclusions are that the Mill River did have greater turbidity, water temperature, dissolved oxygen, and pH compared to the Lower Hop Brook. and the Lower Hob Brook had greater density of macroinvertebrates that were slightly more sensitive to pollution. And all of this is basically what we predicted, except for the dissolved oxygen. That was opposite of what we predicted. We don't know for sure what's causing the differences between these two rivers, but it's pretty likely that land use is probably at least influencing the differences between these two rivers. And this is a really useful comparison to get students analyzing data and putting it into a larger context so they could really see how humans were impacting the Mill River, which is right here next to campus. So, not just humans in general, but Smith College as a community could be affecting the Mill River. And also the mystery of why the dissolved oxygen is higher in the Mill River, which is opposite of we expected, gave us some interesting things to talk about as to how we could figure out what's the reason behind this and thinking about future research and how we could figure that out. I'd like to acknowledge all of the data help that previous Smith College students and colleagues helped collect. This is only a subsample of the students that helped collect the data. And of course, I want to acknowledge Neon for making their amazing data sets available, publicly available, and relatively easy to use. Please reach out to me by email, or you can follow me on Twitter or look at my website. Love to talk to you about using research in classes, using Neon data specifically, anything related, using cures in classes, introductory classes. So happy to talk to you. Please reach out to me. I will also be at the live discussion. Thank you for watching my talk.